anytime we see a patient, we're always thinking about clinical trial opportunities. And that starts from the very beginning when we send patients tumor tissue for genetic analysis. Anytime you do genetic testing on a tumor, it's like looking for a needle in a haystack. And although we test for 300 or more different genetic alterations, sometimes we only find one or two. In cases where there are drugs that can block those alterations, we try to prioritize based on agents that are in clinical trials with drugs that might cross the blood-brain barrier. So an example would be a genetic alteration called BRAF mutations. And BRAF mutations are commonly seen in melanoma, but there are 2 to 3% of patients with high-grade gliomas that have BRAF mutations. And we have patients who we've put on these medications and they've had shrinkage of the tumor. So it's that type of opportunity that's very exciting, and so we're constantly looking for these subsets of patients. So there are other examples such as tumors that have FGFR amplification that might respond to a drug that blocks that signal. We also are really interested in finding tumors that might be more susceptible to the effects of immunotherapy. And so there's a hypothesis that tumors that have many, many mutations, hundreds of mutations, might be more sensitive to some of the drugs that work so well in melanoma, the checkpoint inhibitors. So we have clinical trials for patients that have those, what we call hypermutator phenotype.